I'm Bart Rose. I am a assistant professor of surgery and chief of HPV surgery at the University of Alabama in Birmingham, and I will be speaking about population disparities in neuroendocrine tumors. I have some funding through the NIH and pNETs on this topic. For those of you who aren't familiar, neuroendocrine tumors are derived from ectodermal multipotent stem cells. They can produce aberrant physiology due to hormone secretion, but about 40 to 60% of these tumors will not secrete any measurable hormone. About half of patients will present with metastasis, liver being the most common site. And the natural history of these tumors is variable. Some behave very aggressively and others are very indolent. They can be associated with certain genetic syndromes such as MEN1, von Hippel-Lindau, TSL, and neurofibromatosis 1. The most common sites vary on region. In the US, the most common site is the rectum, followed by the small intestine, and the pancreas being the third most common site. These tumors, as mentioned before, can be functional, meaning that they secrete a hormone. This is more common with pancreas tumors. Depending on the type of hormone secreted, they can have variable symptoms. If they, for example, produce gastrin, patients will often present with recurrent ulcers. Small bowel neuroendocrine tumors and pulmonary neuroendocrine tumors can produce a syndrome called carcinoid syndrome, which is associated with facial flushing, diarrhea, bronchospasm, and heart valve disease. This is usually a sign of metastatic disease, as most of these are processed in the liver. 90% of these patients will have an elevated metabolite of serotonin in their urine, and as mentioned before, more commonly associated with the lung and the midgut. These functional tumors have varying malignant potential. Most of them have the ability to metastasize, with the exception of insulinomas, has a very low risk of metastasis. Non-functional tumors, which are about half of these tumors, typically will present because of the mass effect of the tumor itself, leading to abdominal pain, weight loss from gastric outlet obstruction, or jaundice from biliary obstruction. Usually noted on a CT scan or MRI, but occasionally an endoscopic ultrasound. Biochemical tests can reveal hormone secretion, but this is challenging if there's no clinical syndrome. You can use alternatively a chromogranin A level, although this is variable and can be increased, leading to false positives with any number of medications and sometimes diet. A gallium-68 dotatate scan can be helpful for localization. The incidence of neuroendocrine tumors has been rising. There's been a over six-fold increase in neuroendocrine tumors that have been diagnosed since 1973. There's about 7 per 100,000 persons will be diagnosed with a neuroendocrine tumor in the United States this year. This increase has been across all sites and stages. Interestingly, this occurs at about the same time we see an increase in CT scan use, suggesting that what we're seeing here is actually an increase in incidental tumors, questioning whether or not they have any clinical relevance. NETs have been increasing mostly in the low-grade and localized neuroendocrine tumor variety, again suggesting that we're just catching early, potentially benign neuroendocrine tumors. However, there has been a slight but steady increase in the rest of them over time. There are different types of neuroendocrine tumors. We typically will grade these according to level of differentiation, and then on proliferation is measured by either Ki-67 or mitoses. Very low-grade tumors are well differentiated with low proliferative indices, but as we increase the level of differentiation until they become poorly differentiated, we then refer to these tumors as neuroendocrine carcinomas rather than neuroendocrine tumors, and these are treated much more similarly to other solid tumors and are treated with systemic chemotherapy rather than hormonal therapy or surgery. In 2007, there was this recategorization to include this new category of a well-differentiated but still high proliferative index neuroendocrine tumor called a grade 3 neuroendocrine tumor. The clinical treatment for this classification is yet to be fully hashed out. It is clear, though, that these grade 3 neuroendocrine tumors do behave differently compared to their neuroendocrine carcinoma counterparts who have a very poor median overall survival, and those with a grade 2 well-differentiated neuroendocrine tumors which have an improved overall survival, with this new classification somewhere in between. Now, for a localized non-metastatic neuroendocrine tumor, surgery is often considered curative. We can see here when looking at patients who undergo surgery versus any type of resection of a pancreatic neuroendocrine tumor, there's a significant improvement in survival. However, the question is, could this be due to a selection bias, and that we're selecting patients who have more favorable tumor biology and not operating on those who aren't? This next study tried to answer that question in 2009, where they looked at patients who were resected, patients who were recommended resection, so theoretically were healthy enough to have a resection, but then ultimately opted to not have surgery, and those whose surgery was not recommended. And we can see here again a clear improvement in survival in those patients who had resections compared to those who were recommended resection but did not undergo it, again suggesting that surgery is potentially curative and is a mainstay of treatment for localized disease. Now, there are known racial disparities across many different cancer types, and this is true also in neuroendocrine tumors. However, given it is a relatively rare cancer, this hasn't been studied to the extent as it has in some other malignancies such as breast, prostate, and colorectal. We do know that Black patients have a 11% higher age-adjusted death rate for all cancers compared to white patients. And this is thought to be resulting from a confluence of both socioeconomic, health system, environmental, and biologic factors. Little is known in neuroendocrine tumors. This first study looked at the SEER data set from 1974 to 2015. This included 53,000 patients, about 15% were Black. And what they found was that rural, uninsured, unmarried men with advanced disease were more likely to have poor disease-specific survival. Black patients were found to have worse overall survival, but not disease-specific survival, as noted here, with a hazards ratio of 1.16 for non-Hispanic Black or versus non-Hispanic white. However, the problem with this study was that it was heterogeneous and included both pulmonary and gastrointestinal pancreatic neuroendocrine tumors. This additional study looked at the SEER data set again from 2004 to 2015 this time and excluded pNETs. So this was really focused just on gastrointestinal neuroendocrine tumors. This included a little over 26,000 patients, about 20% were Black and 15% were Hispanic. And they found that white patients were more likely to have surgery, but Black patients had better overall survival and disease-specific survival. Again, this was a heterogeneous neuroendocrine tumor population, including rectal, small bowel, and gastric. The first study to really look at overall survival specific to pNETs looked at the NCDB from 2004 to 2015. It's incorporated about 16,000 patients, of which 12% were Black and 9% were Hispanic. And they found that there was no difference in overall survival by race. Instead, they found that income, education, and insurance type was associated with poor overall survival. This would suggest socioeconomic influence. However, another study looking again at the SEER data set from 2004 and 2013, this time focused specifically on pancreatic neuroendocrine tumors, had a smaller number of about 3,800 patients, of which 13% were Black. But this time found that Black patients were more likely to have distant disease, 29% versus 23% than white patients, were less likely to be offered surgery, and had a worse disease-free survival for local disease, and close to a worse disease-specific survival. However, the p-value was just barely positive. So this would suggest potentially an access issue. Patients were not being offered surgery as often for something that was potentially curable for local disease. When patients did have a resection, their survival was similar. The USNet study group, which was a consortium of a number of high-volume institutions focused on neuroendocrine tumor care, decided to look at their own granular data set, incorporating patients from 2000 and 2016, of which there were nearly 1,200 patients, all of which had had resections for low-grade, well-differentiated neuroendocrine tumors. About 14% of these patients were Black. Within this data set, they found that Black patients had a better 5-year recurrence-free survival, but were more likely to be lymph node positive, 47% versus 36%. Interestingly, we find that white patients and Black patients, if they were lymph node negative, Black patients did significantly better. However, when lymph nodes were positive, recurrence-free survival were similar. This would suggest that Black patients had earlier lymph node metastasis. However, if they received resection before lymph node metastasis occurred, they would have improved survival. This led to our institutional study, looking at our own data set of 151 patients who underwent resection for low-grade, well-differentiated neuroendocrine tumors between 2010 and 2019. This cohort had about 25% Black patients. What we found was that Black patients had larger tumors on average, but did not find a difference in disease-specific survival by race. Patients with greater than 2-centimeter tumors had worse disease-free survival. However, this was not, when we looked at this by race, we didn't find a significant difference in recurrence-free survival based on tumor size. So this would suggest that, well, our Black patients had larger tumors, it didn't have the same impact on survival as it did on our white patients. It began to suggest there may be a biologic influence on outcomes, both on the USNet study group, which suggested a difference in lymph node metastasis, and on our data set that suggested a difference in size was not translating to a difference in outcomes. Now, however, this study was relatively underpowered. And all of this really plays into trying to figure out who should be offered resection. So we think that resection is potentially curative and definitely has an impact on survival. Currently, recommendations and guidelines will recommend that patients who are symptomatic with either functional tumors or non-functional tumors that are large and causing symptoms such as pain or obstruction should go undergo resection. However, there's some controversy about what to do with asymptomatic patients who have non-functional tumors. Current guidelines recommend observation for tumors less than 1 centimeter, resection for tumors greater than 2 centimeters, and for clinicians to use clinical judgment for tumors that are sized between 1 and 2 centimeters. The size we use as a proxy for likelihood of metastatic disease. This is based on a number of studies, which in 2001, this meta-analysis was performed that showed that size was correlated with risk of lymph node metastasis. We know that when tumors in the pancreas, pancreatic neuroendocrine tumors get larger than 2 centimeters, there's up to a 40% risk of lymph node metastasis at time of resection. Well, sub 2 centimeter tumors have somewhere between a 5 to maybe 10% risk of lymph node metastasis, suggesting that observation may be adequate. However, all of these data sets were derived from primarily European and white patients and had not been validated uh, based on patient race. This led to our recent paper from our group in the Journal of American College of Surgeons, where we investigated whether or not current size-based guidelines in Black patients with pNETs were accurately predicting risk of lymph node metastasis. We performed a retrospective analysis of the USNet study group combined with our institutional data. This incorporated patients with resected tumors from 1998 to 2019. All these patients had non-functional, well-differentiated neuroendocrine tumors that were not associated with an inherited genetic syndrome. This was then validated against the larger National Cancer Database, or NCDB, data set. We excluded patients who had known metastatic disease, who had resections that were done for non-curative intent, and all high-grade or poorly differentiated neuroendocrine tumors. These were the demographics of both the study set and the validation data set. And to summarize this, essentially the findings that we found were that Black patients were more likely to be younger, female, and have lymph node positive disease than their white counterparts. The size characteristics at baseline were similar between the two patient groups. When we looked at the risk of lymph node metastasis based on size of the primary tumor, we found that first, for greater than 3 centimeter tumors, there's no statistical difference in the USNet study group or in the NCDB data set. Risk of lymph node metastasis was somewhere close to 40%, which is in line with the previously published meta-analysis. When we looked at tumors between 2 to 3 centimeters, we again found no difference in the USNet study group or in the validation set in the NCDB. However, when we looked at sub 2 centimeter tumors, now these are tumors that we would recommend observation because they theoretically have a very low risk of lymph node metastasis, we found a stark difference. First, in the USNet study group, only 5% of our white patients had lymph node metastasis time of resection, which is in line with published data. But up to 23% of Black patients had a lymph node that was positive with a sub 2 centimeter tumor. In the NCDB data set, this difference was a little bit less stark, but was still significantly positive with a 2 times risk of lymph node metastasis for Black patients compared to white. When we looked at that sub 2 centimeter subset even closer, we found that the risk of a sub 1 centimeter tumor was nearly 0 in white patients for risk of lymph node metastasis, but up to 25% in the USNet study group. And in the 1 to 2 centimeter range, it was 7% versus 23%. In the NCDB, this was a little bit less stark, but remained significant. Again, having a nearly 2-fold change in risk of lymph node metastasis in Black patients compared to white. And the study was somewhat limited as it is a retrospective review and it introduces bias by nature. Individual patients may be represented in both data sets, as all of the USNet study group consortium sites also contribute to the NCDB. And the NCDB does not report on tumor grade, but it does report on differentiation. So therefore, we should be able to eliminate poorly differentiated tumors, which are generally high grade. From that study, we concluded that recommendations for surveillance of 2 centimeter pancreatic neuroendocrine tumors are based on a low rate of lymph node metastasis seen in predominantly white patient population, that the incidence of lymph node metastasis in Black patients in sub 2 centimeter tumors is clinically relevant and concerning, and that current guidelines may not be universally applicable, and a more aggressive approach towards resection in Black patients with small pNETs may be warranted. Based on our clinical data, it would suggest that there is underlying differential biology driving some of these disparate findings between our two racial groups. The question goes to, what do we know about the genomics of pancreatic neuroendocrine tumors? Well, we know that the US population is about 13% Black. But when we look at analysis of racial and ethnic representation in all cancer genomics, we see that only about 2% of sequenced genomes of tumors are from Black patients. This led us to ask the question, what is known in pancreatic neuroendocrine tumors specifically? To answer this question, we performed a meta-analysis and review using natural language processing. We started with 3,300 records from PubMed and Embase. And after excluding articles for duplication, not meeting inclusion criteria, or not reporting the race or ethnicity of the patient subjects, we were left with 13 articles to be included for a quantitative analysis of representation. This work was primarily done by a graduate student, Brendan Herring. When we looked at these 13 articles, we found that 9 articles included analysis of tumor DNA. 141 patients were included in that. 3 articles focused on the transcriptome, including 79 patients, and only 1 article looked at methylation status, and that included 40 patients. We looked at these genomic studies and broke them down by race. We found that only 26 patients were included in all 1,938 sequenced patients, representing about 1.3% of the total. As there were no published data sets looking at the differential expression between Black and white patients, we looked for ways to answer this question. We found the AACR Project Genie dataset and utilized this as an attempt to begin to understand where there were differential expressions between racial groups of common genes. The AACR Genie dataset includes a number of panels of commonly mutated cancer genes. When we looked at in that gene dataset and looked specifically for mutations that were likely to lead to a functional loss of proteins such as indels and truncations, we found that 337 white patients had pNETs for analysis and 23 Black patients. When we look for differentially expressed genes between the 2 data sets, we found a number of differentially expressed genes. An interesting one was the MEN1 gene, which has been shown previously to be the most mutated gene in pancreatic neuroendocrine tumors in about 40% of tumors, which is in line with what we saw in our white patients. Interestingly, our Black patients had nearly a half of that their tumors were mutated with MEN1 at 17%. We found additional genes that were enriched within our Black patient population that were not within our white. We then asked the question as whether or not this mutation in the MEN1 and the other commonly mutated genes of DAX and HRAX translated to the protein level. And while we found that there was no statistical difference in the staining for DAX and HRAX on a tissue microarray of patients at our institution with resected pNETs, this included 40 white patients and 13 Black patients, we did find a statistically different number of patients with MEN1 mutations leading to a loss of protein, with 11 of our white patients and none of our Black patients having mutations in MEN1 leading to a loss of function. Because perhaps the most variation between racial groups occurs at the epigenetic level, and such differences in epigenetics have been shown to influence racial disparities in multiple cancer types, we took particular interest in the epigenetic regulatory networks that were mutated. We conducted clustering and gene ontology analysis to figure out if and how these genes were related to one another. What we found were these genes formed a nice interaction network with one another. Clustering brought in other genes that were significantly associated with our query genes to augment the network and help determine the pathways that were affected. K-means clustering put these genes into 3 distinct functional interaction clusters depicted by colors here. MDM2 degrades p53. MYOD1 is involved in muscle cell differentiation, but in cancer plays a role in suppressing migration and invasion. SIRT1 inhibits apoptosis and senescence and supports the viability, proliferation, and invasion of pancreatic cancer cells. SMAD2 is a tumor suppressor. SMARC-C1 is another core subunit of the Swift complex BAF. And lastly, KDM6A is a histone demethylase. Looking at the pathways that these genes affect through their interaction network, we pulled out four major categories. In red is endocrine function and pancreatic beta cell differentiation, which you would expect with pancreatic neuroendocrine tumor. Yellow is chromatin remodeling and nucleosome regulation. Green is proliferative signaling. And purple is p53 and DNA damage response. Using this information, we wanted to see if we could characterize some specific functional convergences in the genes that we identified in original analysis. Because we are including immunohistochemistry and mutational data in our review of racial groups and GEP-NET sequencing, we started to build a rudimentary model of them. Interestingly, KM2TD is necessary for menin to exert its function as a histone methyltransferase that methylates lysine 4 on the H3 tail, promoting transcription of a host of factors including CDK inhibitors P21 and P18, among other anti-proliferative genes. SMARC-CB2 and KM2D and EP300 form a complex with one another that increases lysine 27 acetylation and enhancer elements throughout the genome, regulating a complex gene network. EP300 and Swift complex also interact with one another to facilitate the enhancer function of the endocrine transcription factor PDX1, which is key in pancreatic beta cell differentiation and maintenance of differentiation states in the adult pancreas. Importantly, loss of PDX function has been shown to confer poor prognosis in pNETs, with the enhancer signatures clearly influencing pNET biology. Finally, MEN1, SMARCB1, and KMT2D all inhibit the nuclear translocation and activation of active beta-catenin, thereby inhibiting the pro-proliferative signals activated through the Wnt pathway. These data indicate that perhaps while Black patients have fewer mutations in MEN1, the genes that are mutated in its stead seem to share some important functional roles with it. Hence, the aberrations may simply lie at these different nodes in MEN's functional pathway and ultimately result in similar downstream effects in these cancers, while conferring some differences in the tumor biology that we're seeing at the clinical level, namely differences in the rate of lymph node metastasis. In summary, significant differences exist in the clinical outcomes between Black and white patients with pNETs. Black patients are more likely to have early lymph node metastasis. The MEN1 gene is commonly mutated in white patients, but apparently less so in Black patients. Other epigenetic regulators are more commonly mutated in Black patients, so may have the same downstream effect. I'd like to acknowledge my collaborators, Herb Chen, Shishir Maythel, and Ryan Fields, and Clayton Yates, as well as the lab members who performed the majority of this work: Brendan Herring, candidate MD PhD, Rachel Gunter, postdoc in my lab, Rajang Paiwal, former postdoc, and Amanda Fang, a medical student who is now in her residency. Thank you.